Welcome to BioBiz Buzz. I'm Mike Ward. I'm the Global Head of Life Sciences and Healthcare Thought Leadership at Clarivate, which is a leading global provider of transformative intelligence, offering enriched data, insights, and analytics to the life sciences and healthcare sectors. I'm also one of the regular hosts of BioBiz Buzz, a podcast that seeks to speak to business leaders at the cutting edge of the life sciences. And today, we're diving into one of the most transformative technologies in modern medicine, CRISPR gene editing. So imagine a world where genetic diseases like sickle cell anemia or beta thalassemia aren't just managed, they're actually cured with a single treatment. And that world is now a reality. In late 2023, the FDA approved Caszyme, the first CRISPR-based therapy, making a seismic shift approaching previously untreatable conditions. And this is just the beginning. CRISPR's precision in cutting and repairing DNA isn't just rewriting genetic code, it's rewriting the playbook of the entire pharmaceutical industry. CRISPR is unlocking doors we didn't even know existed, from turbocharging drug discovery to enabling personalized cancer therapies. In this episode, we're diving into this cutting-edge world of CRISPR gene editing with Dr. Monika Paule, CEO and co-founder of Caszyme, which is a Lithuanian biotech company at the forefront of developing next-generation molecular tools. Since its founding in 2017, Caszyme has become a key player in advancing CRISPR-Cas technology for applications ranging from therapeutics to sustainable agriculture. So please sit back and listen as we explore how Monika and her team are shaping the future of genetic engineering, the challenges of commercializing breakthrough science, and her vision for a more inclusive biotech industry. So Monika, let's start with the Caszyme origin story. What inspired you to co-found a CRISPR-focused company in Lithuania?
CRISPR is quite well established in Lithuania for, almost 20 years. And my scientific co-founders, Professor Virginijus Šikšnys and Dr. Giedrius Gasiūnas, they started to work with CRISPR something like 18 years ago. And Dr. Giedrius Gasiūnas, our current CSO, he worked with CRISPR all his life, from early bachelor studies and proceeded into master's and doctoral degree. And they did this first discoveries in the CRISPR-Cas9 field and claimed one of the first patents as well as scientific articles. And throughout the years, they kept working on this technology and realized together with some of our commercial partners that this technology, apart of being very academic and fundamental, also has a lot of applicational potential. And that's where the idea of Caszyme, a commercial entity, but at the same time, CRISPR expert, working with multiple industries and multiple partners, came into our minds.
So you mentioned there that Caszyme has this sort of focus on multiple industries. So can you sort of describe how Caszyme's approach to gene editing differs from, for example, other companies in the space?
Yes. So from the very beginning of Caszyme, which was 8 years ago, we saw CRISPR gene editing as a technology that could enable different industries, different companies, different business lines, different products. And our idea was through our knowledge, through the research and discovery that we did, through identifying novel nucleases, to work with all of these different industries and companies and make the technology as accessible, as available as possible, and also to help all the complex technical issues that companies pursuing end products face. That's where, we as CRISPR experts can come in and help them.
So how do you differentiate, though, what you're doing in the CRISPR field with, for example, the other companies who are in the space? What's your secret sauce?
Yeah, so our secret sauce, first of all, is the expanded gene editing toolbox that we have developed. So initially, as everybody, we focused on CRISPR-Cas9, but in recent years, we moved to the Cas12 field and developed our own proprietary nuclease, Cas12L, where we proudly say that L stands for Lithuania, where we are coming from. And the approach is to find nucleases at the same time could be easily reprogrammable and, constantly developed using AI and machine learning to fit in as many different applications as possible. So let's say we solved the diversity of PAMs needed initially with our Cas9s. Now, when we have this Cas12L, it's very comparable to Cas9, but at the same time could easily replace it and also solve the issues like, let's say, size limitations in the therapeutic space where, you need a small nuclease. So yeah, we have a pretty small nuclease, but at the same time, as I said, easily comparable and exchangeable with Cas9, which enables more companies to access the field and, like different characteristics that industry needs, like temperature tolerance or specificity, efficiency, activity in human cells. So all of these issues we solve step by step and really make the very accessible tools. Whereas I think the whole industry initially was moving, okay, I have my own unique nuclease, and then I do one kind of a product, one kind of a therapy with it, but don't have this more like open approach where one tool can be used by, let's say, multiple players in the industry.
So how big is therefore that toolbox that you've created?
So we have more than 80 different Cas9 variants and way less Cas12L variants. But as we said, we'd really then put this new approach of AI and machine learning engineering and have only four different variants, but very good ones.
And sort of, in terms of efficiency, how has that improved?
So now we see that Cas12L on different targets can have a very similar editing efficiency to Cas9. And let's say through the activity perspective, so in certain targets, we see even up to 100% editing efficiency. And so that's kind of really an impressive numbers, we think.
I mean, one of the other interesting, technologies that has emerged in sort of the past 5 years or so, certainly in terms of sort of clinical utility, is mRNA. And I believe that you guys are using or are marrying the sort of the two technology platforms. So how is that panning out?
So in case of mRNA, we saw like a natural need from our therapeutic partners to also have this capability within our company. So let's say at the very early stage of optimizing mRNA and combining it with gene editing. So we also built in this capability in-house just to actually serve our partners in the full scope, as well as, we showed the Cas12L delivery compatibility with different methods. So let's say either AAV or LNPs or the same mentioned mRNA. So, now we can claim that we definitely see the full compatibility and delivery with all these different approaches of our one tool. So that's kind of amazing.
So what you've done is you created a platform that is of potential use to sort of partnerships or collaborators. So could you just sort of explain how those collaborations are helping you move forward?
Absolutely. So we have different collaborations in different industries and quite well established ones. So let's say in case of the therapeutics, so probably our most well-known collaboration is with the Spanish company Integra Therapeutics, who licensed our Cas12L technology and combines it with its TCAT gene writing platform. And they are aiming for rare pediatric diseases. So we started working with them already quite some years ago, and they believed in the tool, and we engineered it on the way, also trying to understand the needs of their therapeutic applications. So now they have a very well functioning tool, and we looking forward for them to generate clinical data and to show that our tool works in the clinic. Another example is, let's say, in the diagnostics field, where we just recently announced a partnership with Korean company NSage and Vilnius University from Lithuania on CRISPR application in diagnostics field, where we as free partners bring our technologies together and seek to develop also like universal diagnostics platform for humans, but not only for animals and even agriculture as well.
Okay, so because normally sort of gene editing or CRISPR is often associated with rare diseases, so there are other applications, diagnostics, and as you're sort of saying, in places like agriculture, etc. I mean, what can we expect gene editing to deliver in those spaces?
So I think now we see most of the publicity of CRISPR gene editing in the therapeutic field. And that's completely fine because, we are humans and we talk about what affects us the most. But as you said, CRISPR gene editing and we Caszyme have interest also in the diagnostics field, where gene editing is very well applicable. We just need to solve certain technical issues. But at the same time, agriculture field. So in the agriculture field, we are building some new partnerships, which we will announce pretty soon. But also we have a very long-term collaboration with Corteva AgriScience, which is a leading agri-tech company having a very strong portfolio of patents and discoveries in the gene editing field. And we've been collaborating with them already for 8 years. So to develop, novel traits and solve certain disease and efficiency issues in the field. So I think in this current climate change issue, CRISPR gene editing application in the agriculture becomes very important as well.
So I'm saying CRISPR, I mean, as you mentioned in one of your earlier answers, it's a new technology, certainly from sort of the first discoveries was sort of, I guess, like 20 years or so. What challenges, when you're sort of, at the cutting edge, what challenges does a company like Caszyme face when sort of like developing CRISPR tools, for example?
So I think one of the challenges, which we are very good in solving in, is the general complexity of CRISPR as a technology, all the technical challenges that all of the labs face all over the world, but that is pretty well solvable. Another issue is IP, because there's a lot of competition instead of collaboration. So we really tend to spread the idea that maybe we could collaborate more and find the space for everybody, because the more companies are pursuing CRISPR gene editing, the more amazing end products we will have in all these different industries that we have mentioned. But also, let's say, looking into the therapeutic field, of course, all the regulations is quite a challenge. And if you want to build a therapy based on gene editing, you have to show all this preclinical safety, efficacy data, and it's not so straightforward. So this is quite a challenge, but I believe also we as Caszyme are pushing some of our pipelines ourselves to show how well our tools work, as well as the partners we are working. We will show some amazing data even this year that will enable the path of our Cas12L into clinics.
All right. And as we sort of draw to close, I'd actually sort of, maybe sort of pivot to a more personal challenge. As a female CEO in biotech, what barriers have you faced, and how can the industry sort of improve in terms of sort of the diversity of its leadership?
So I would say it's a very male-dominated industry, but as many others. And although, I mean, I'm more than 10 years in this field and almost 20 years in the biotech in general, I see the challenges, first of all, being a young person and being a person, first of all, driven by the business mindset. But at the same time, I find it very rewarding to see how, in general, the industry is changing, how it is diversifying, not only from the gender perspective, but just from, where people are coming from and, what kind of amazing things and added value can be achieved in all these collaborations. But at the same time, yes, I would say I had to work very hard all my life and really to prove myself. But yeah, that's probably what you have to do if you want to be successful, as it is said. Because I always say that the secret of success is very, very hard work and belief in what you are doing.
Right, sure. And sort of as we do draw to a close, looking ahead, what's next for Caszyme? I mean, you mentioned that, maybe there's some sort of deals in line. You've just done this diagnostics consortium that you created. So what sort of milestones can we look out for?
So we as Caszyme now build our business on 3 pillars. So we continue our R&D efforts, both in different partnerships, to build novel tools, to enhance existing gene editing portfolios, and at the same time to have our own CRISPR gene editing solutions. We continue developing our platform, generating more amazing data, first of all, on Cas12L and making it available through licensing to many companies, especially in the therapeutic fields. And we are pursuing our own pipeline in therapeutics based on our Cas12L. At the same time, as I said, to show how amazing this tool is, but also to solve some unmet needs that are coming from the hospitals that want to collaborate with us and companies and so on, and also in the diagnostics field. So that's our, let's say, short and midterm goals to grow our business on these 3 pillars.
Right, great. So, Monika, thanks very much for taking time today, sharing insights that I found both informative and inspiring. So what we've learned today is that CRISPR isn't just about treating rare diseases. It has the potential to find ways to treat devastating diseases that affect much larger patient groups. Moreover, it's revolutionary actually how we might develop more sustainable and enhance industrial processes. Of course, as you've mentioned, challenges remain, delivery hurdles, there are ethical debates, and also just basically the cost of these new therapies such as Casgevy, remind us that CRISPR isn't just a sort of a magic bullet. So the momentum is undeniable. We're seeing some of the big industry giants getting into the space. So it looks like it's pretty exciting times for you and for us. So thanks very much, Monika.
Thank you.
So I'm curious how you, the audience, have felt about the conversation. Please send us your feedback. If you want to stay tuned for more editions of BioBiz Buzz, follow our LinkedIn page where we will post alerts for future episode releases. So in closing, I'd like to thank Monika again for joining me. I'm Mike Ward, and I'll hope you'll join me for the next episode of BioBiz Buzz. Goodbye.